Dear student, I am Dr. Prakash Mungli, Professor in Biochemistry. So in this video, I will be explaining you regulation of enzyme activity. When it comes to regulation of enzyme activity, so regulation can be divided into a short-term regulation and long-term regulation. Now, why we need this regulation of enzyme? So, because our metabolic pathways always constantly they are changing, they will be dynamic because the amount of substrate that will be getting into these enzymes will vary all the time and depending on the physiological situation, physiological condition, so the metabolic pathways need to be altered and that's why enzymes need to be regulated. Certain key enzymes in the metabolic pathways need to be regulated, not all the enzymes. There will be multiple enzymes in a metabolic pathway, not all of them need to be regulated. Only the key enzymes which are regulated enzymes or the rate limiting enzymes need to be regulated so that metabolic pathways can be altered according to the physiological need of the cell. So how these enzyme activity is regulated? So it can be regulated on a short-term basis or short-term regulation indicating that only the available enzyme or the existing enzyme activity can be changed. It's like fine tuning the enzyme, fine control of the enzyme. We also refer it as a qualitative control. So in short-term regulation, we have allosteric regulation where the allosteric modulators will keep the enzyme activity high or low, or it can be covalent modification where addition of certain things like phosphate over the enzyme's surface may alter the enzyme activity, keeping it either active or inactive. So this kind of fine control where enzyme concentration is not changed, but it is the existing enzyme's activity is kind of altered, and that is called as short-term regulation. Coming to the long-term regulation. Long-term regulation is alteration in the concentration of an enzyme. It's a course control mechanism where a quantity of an enzyme is altered accordingly, either increase in the quantity of enzyme or decrease in the quantity of enzyme. So that means when we are changing the quantity of an enzyme, that means in the cell, the gene has to be regulated. Means the DNA that is coding for mRNA that is coding for that particular enzyme need to be altered. So if there is need to increase the enzyme concentration, to increase the enzyme activity there, to make more and more products, induction has to go on. So induction increases the gene transcription for the enzyme. So enzyme concentration will increase. Whereas repression, in repression, there will be gene inactivation. Basically, gene will be repressed where it will be methylated so that gene expression is not going on, mRNA is not coming sufficiently. That means protein, that is enzyme, is not synthesized sufficiently. That will decrease the enzyme activity. So basically, it's a course control mechanism. Now, the rate of reaction of most enzymes are responsive to changes in the substrate concentration. Intracellular levels of many substrate is in the range of Km, because Km is the substrate concentration at which enzyme reaches off the maximum velocity. So the substrate concentration of many of our intracellular enzymes, usually it will be at the range of that Km, not necessarily at the range where it reaches Vmax, which is a saturation kinetics, but at least it will be there at the range of Km. And many regulatory enzymes, they respond to allosteric modulators or allosteric effectors, and also they respond to covalent modification. Thereby, the activity of enzyme can be altered without changing the concentration of an enzyme. And if there is any need for changing the concentration of enzyme according to the physiological condition, that can also be done. So it's a fine control and the coarse control that is going to regulate enzyme activity. Next. Now let's look at allosteric regulation. Allosteric enzymes are oligomers. They are having multiple subunits. Allos means other. That means not only allosteric enzymes have got active site, they also got other site other than the active site referred as allosteric site. And this is where allosteric effector will go and bind. And this allosteric effector it can be positive or it can be negative. So allosteric enzymes have active site and allosteric site, where allosteric effectors will go and bind and work positively or negatively depending on what type of allosteric effector it is. And also note that since allosteric enzymes are oligomers, they have multiple subunits. That means they may have more than one active site, and also they may have more than one allosteric site, because these are multi-subunit enzymes. Now, if you look at the reaction velocity curve for allosteric enzymes, compare that with the simple enzymes which follow Michaelis-Menten kinetics. See, the red line that you are seeing here is the reaction velocity curve for the simple enzyme, and it is hyperbola. It is hyperbolic curve. That means there will be rise in the velocity of the reaction in the initial substrate concentration, and then it is taking a plateau stage there, because it is reaching the saturation. So this type of reaction velocity curve is the hyperbolic feature, which indicates simple enzyme catalyzed reaction. Whereas the allosteric enzyme catalyzed reaction velocity curve is sigmoidal. Sigmoidal curve is the characteristic of allosteric enzymes, or any protein that has got multiple subunits, and like hemoglobin molecule, so will show a sigmoidal curve. So always remember, sigmoidal is indicative of multiple subunits cooperating with one another and giving that sigmoidal curve. So sigmoidicity, it is the phenomenon of cooperativity, remember that. Whereas hyperbolic curve do not show the cooperativity. So simple enzymes, it is showing hyperbolic curve and they follow Michaelis-Menten kinetics, whereas allosteric enzymes, they will show sigmoidal curve, which is the which indicates the phenomenon of cooperativity going on between the subunits. And sigmoidal curve is it is for allosteric enzymes, and majority of the regulated enzymes in our body, rate determining regulated enzymes in the metabolic pathways, they are all usually allosteric enzymes. Next. So allosteric enzymes are regulated by allosteric effectors, and these allosteric effectors can be modifiers or modulators that they bind non-covalently to a regulatory site or the allosteric site. So the presence of allosteric effector binding to an allosteric enzymes can alter Km, it can alter Vmax, or it can alter both. They can increase the Km, decrease the Km, increase the Vmax, decrease the Vmax, or alter both. So allosteric enzymes exhibit cooperativity phenomenon. That means these subunits, they coordinate with one another in a positive way or in a negative way. But there is a cooperativity, and that cooperativity is the one that shows a sigmoidal reaction velocity curve. Now let's look at reaction velocity curve for allosteric enzymes which is a sigmoidal curve. So the curve B here that you are seeing, curve B, is under normal conditions without any allosteric modulators. So the reaction velocity curve is shown, which is a sigmoidal. That's the blue line there. Now whenever a positive allosteric modulator is added, into the system, so this positive allosteric modulator will go and bind to an allosteric site and act positively onto an enzyme. Maybe it will decrease the Km, increase the Vmax and all that. So it is going to shift the reaction velocity curve towards the left side of it. So this is what is the effect of positive allosteric modulator, where the reaction velocity curve, that is the sigmoidal curve, is shifted towards the left side of it. Now if there is a negative allosteric modulator, the reaction velocity curve will be shifted to the right side of it. So let's look at how exactly it affects the Km here. So this is because in this particular figure, Vmax is remaining same, only Km can be altered here. So let's look at that. So in the positive allosteric modulator, whenever the positive allosteric modulator is added, let's see how it's going to change the Km of this particular curve. So let's take the half of this Vmax and put that to this purple color line. That's a reaction velocity curve in the presence of, in the absence of modulator. Then let's write it as Km1. Okay. Now in the presence of positive allosteric modulator, so the F we max here, same line. So bring it down to the x-axis and it is coming and interpolating there in the x-axis substrate concentration. This will be Km2. Okay. Now look at negative allosteric modulator reaction velocity curve. So reaction velocity curve for negative allosteric modulator, let's put that as a Km, which is interpolating here to this reaction velocity curve for negative modulator, and bring that down to the x-axis and let's put that as Km3. Now let's see what happens. Compare that with the Km1. So Km1 is, say, let's put that as number 4 millimoles or something. Now let's look at Km2, that green here. So Km2, it is 2, like it is going towards 0. That means Km is decreased from 4 to 2. In the negative allosteric modulator side, so Km is increasing to 6. So that means in this particular example, so the positive allosteric modulators have simply decreased the Km of an enzyme. Thereby, the enzyme's affinity for the substrate is increased and enzyme is working even at low substrate concentration and making the product. Whereas in the presence of negative allosteric modulator, the same enzyme, because negative allosteric modulator is there, it needs more substrate to reach the same maximum velocity. So the Km is increased. So always remember, positive allosteric modulator will shift the reaction velocity curve to the left side of it from the normal, and the negative allosteric modulator will shift the reaction velocity curve to the right side of it from the normal. Okay, so the negative effectors works negatively on the enzyme, whereas positive effectors work positively on the enzyme. So the negative allosteric effectors shift the reaction velocity curve towards the right, whereas the positive allosteric modulator will shift the reaction velocity curve towards the left. So example for allosteric regulation, examples given here, like allosynthase. Negative allosteric modulator for allosynthase is heme. Sometimes negative allosteric modulator can be written as inhibitor, but otherwise, strictly speaking, it should be mentioned as a negative allosteric modulator and positive allosteric modulator. Activator and inhibitor. So, inhibitor can be confusing because these are our own body molecules that are acting negatively on regulatory enzymes. So, let's consider activator as a positive allosteric modulator, inhibitor as negative allosteric modulator in this particular table. Now, the allosynthase negative allosteric modulator is the end product, that is heme. Aspartate transcarbamylase enzyme, which is involved in pyrimidine biosynthesis. So, the positive allosteric modulator is ATP and the negative allosteric modulator is CTP, cytidine triphosphate. FMGE reductase, negative allosteric modulator is the end product, that is cholesterol. Phosphofructokinase enzyme, fructose 2,6-bisphosphate is the positive allosteric modulator and the negative allosteric modulator is citrate. Pyruvate carboxylase, positive allosteric modulator is acetyl-CoA and the negative allosteric modulator is ADP. Acetyl-CoA carboxylase enzyme, which is the enzyme involved in fatty acid biosynthesis. So, the positive allosteric modulator is citrate, negative allosteric modulator is acyl-CoA, which is the end product here. And carnitine palmitoyl transferase 1 enzyme, negative allosteric modulator is malonyl-CoA. And continuing here, like citrate synthase, negative allosteric modulator is ATP, carbamyl phosphate synthetase 1, positive allosteric modulator is N-acetyl glutamate, which is carbamyl phosphate synthetase 1 is the enzyme involved in urea cycle, and carbamyl phosphate synthetase 2 enzyme, allosteric activator is PRPP, whereas the negative allosteric modulator is UTP. So, PFK1 is the rate limiting and most regulated enzyme in glycolysis, and the positive allosteric effectors for PFK1 is AMP and fructose 2,6-bisphosphate, which will shift the sigmoidal curve towards the left side of it because these are positive allosteric modulators. And the negative allosteric effectors for this enzyme is ATP and citrate, Since they are negative, they are going to shift the reaction velocity curve towards the right side of it. Coming to covalent modification. Now, the process of addition or removal of a group by making or breaking a covalent bond can alter enzyme activity. So, enzyme activity either can be increased or decreased due to covalent modification. So, there can be two types of covalent modification. One is a reversible covalent modification, other is irreversible covalent modification. So, in the irreversible covalent modification, there can be partially proteolysis or zymogen activation, where the proteolysis happens and a protein is totally made different. It's an irreversible process. Whereas in a reversible process, addition or removal of particular groups can happen, like addition of phosphate or something. So, common methods of reversible covalent modification is phosphorylation and dephosphorylation. This is in fact the most common covalent modification in our cells, phosphorylation and dephosphorylation process. Other covalent modification can be addition of methyl group, that is methylation, addition of ADP ribosylation process, adenylation process, acetylation process. All these things can modify an enzyme activity. Like example for acetylation is, so this is about covalent modification. So that's all about regulation of enzyme activity. So here we have seen short-term regulation and long-term regulation. In short-term regulation, we have seen allosteric modulation and covalent modification. In long-term regulation, there will be induction and repression, thereby changing the concentration of enzyme itself. So that's all about the regulation of enzyme activity. Thank you.